Cellular respiration is the process in which cells derive energy from glucose. There are three main steps of aerobic respiration. These include glycolysis, shared with fermentation, the Krebs cycle, and the electron transport chain. Fermentation is anaerobic or doesn't need oxygen, unlike aerobic processes. Glycolysis is the breakdown of glucose molecules. Glycolysis is one method cells use to produce energy. Glycolysis does not need oxygen. Glucose is taken out of things we eat. For example, this piece of toast. We are going to follow this singular glucose molecule through each of the steps of both aerobic and anaerobic respiration to derive ATP. The equation for cellular respiration is as follows. Glucose and oxygen make the products, which are carbon dioxide, water, and ATP. During the first step of cellular respiration, glycolysis, glucose is located in the cytoplasm and is anaerobic. 2 ATP are used to start the step, and glucose is turned into 2 net ATP, 2 pyruvate, and 2 NADH. Here, ATP is produced via substrate-level phosphorylation, which means the phosphate group is directly transferred from a high-energy substrate to ATP to form ATP. Nicotinamine adenine dinucleotide is a coenzyme central to metabolism. It is found in all living things. It has the ability to transfer electrons. The pyruvate passes through the outer mitochondrial membrane and into the inner membrane, turning into acetyl-CoA when the pyruvate is oxidized. Carbon dioxide is released and 2 NADH is produced. During the Krebs cycle, a process that requires oxygen, the acetyl-CoA is turned into 2 ATP, 6 NADH, and 2 FADH. FADH, or flavin adenine dinucleotide, is a redox-active coenzyme, which is involved in several enzymatic reactions in the metabolism. FADH assists in transferring electrons. ATP in the Krebs cycle is also produced by substrate-level phosphorylation, where a high-energy phosphate group is transferred directly to ADP from a substrate molecule. The final step, the electron transport chain, is a key component in oxidative phosphorylation and chemoosmosis. This step is aerobic. NADH changes to NAD+ when it releases its electrons into the first part of electron transport chain, pumping hydrogen ions into the inner membrane space to create a proton gradient. The electrons are moved. Picking up electrons from FADH, changing to FAD, while electrons move, more hydrogen ions are transferred into the inner membrane space, building up a high concentration of hydrogen ions. As shown here, oxygen accepts the electrons and turns into water. ATP synthase, which is located in the cristae of the mitochondria, utilizes high concentration of hydrogen ions to create ATP by adding phosphate to ADP. The cristae are the folds in the inner mitochondrial membrane where ATP synthase is located, and where much of the ATP production occurs during the oxidative phosphorylation. This is called oxidative phosphorylation because the process relies on oxygen in the electron transport chain to produce ATP. Overall, aerobic cellular respiration produced about a net total of 38 ATP molecules. The second type of cellular respiration is fermentation. Fermentation has the first step of aerobic respiration but differs after. There are 2 types of fermentation. These are alcoholic and lactic acid. Fermentation needs to restore NADH to NAD+ without oxygen. In alcoholic fermentation, the 2 pyruvate made in glycolysis are used to produce carbon dioxide into ethanol. Acetaldehyde, as shown here, is represented by my hand, allows 2 NADH to be oxidized into NAD+. This process also includes a substrate-level phosphorylation, where a phosphate group is transferred directly from a substrate molecule to ADP, forming ATP. In lactic acid fermentation, after glycolysis, 2 pyruvate are still taken, but they are used as an electron acceptor similar to acetaldehyde, allowing 2 NADH to be oxidized into NAD+. Substrate-level phosphorylation occurs here, generating a small amount of ATP. The net total of ATP produced in alcoholic fermentation is around 2 ATP per glucose, along with lactic acid. As you can see, fermentation produces significantly less ATP compared to aerobic respiration at 38.